Hello everyone, welcome back. In this video, let us discuss about the different types of antigens. Species-specific antigens, isospecific antigens, autospecific antigens, organ-specific antigens, heterogenetic or heterophile antigens. These are the different types of antigens. Now let us discuss about each of them in detail. Coming to the first one, that is species-specific antigens. Tissues of all individuals in a species. For example, if you take one species and all the individuals which belong to this species will have this species specific antigens, and there exists some degree of correlation between those antigens in a related species, not between the unrelated species. There will be a degree of correlation between these antigens of related species. This immunological relationship parallels phylogenetic relationships. These antigens have been of assistance in tracing evolutionary relationships and also have some applications in forensic science. If we come to know the presence of this species-specific antigen in so and so species, and if we identify a new species, we can relate whether it belongs to this group or not, based on the presence of this species-specific antigens. That is what I mentioned. It is useful in tracing evolutionary relationships and also have some applications in the field of forensics. Coming to the other type, that is isospecific antigens. These isospecific antigens are found in some but not in all the members of this species. And the species may be grouped based on the presence of the different isospecific antigens. Best example is human erythrocyte antigens. So here individuals can be classified based on the different blood groups, right? Some people will have A group, some will have B group, some will have AB and O group. There are 4 different blood groups, right? If a person is having A group, we can't give him B blood group, right? So these isospecific antigens play a very important role during blood transfusion. Moreover, isoimmunization during pregnancy, and they also provide valuable evidence in disputed parentage. Coming to the other type, that is autospecific antigens. Sequestered antigens that are not normally found free in circulation or tissue fluids. Antigens that are absent during the embryonic life and develop later are not recognized as self-antigens. See here, these antigens are not present during the embryonic stage, but they develop later. Once they develop and once they are released into the tissues, what happens? Our body will recognize them as foreign particles. They are not considered as self because they are not present at the embryonic stage and they develop later. So what happens? Antibodies will be produced against them. This is what exactly happens with autoimmune diseases. Though they were self, they will be considered as non-self by our body. Coming to the other type, that is organ-specific antigen. Some organs such as brain, kidney of different species has specific antigen. See here, different species. For example, if we take human beings, our organs will have a specific antigen which are different from the other species organs. Such antigens characteristic for an organ or tissue are called as organ-specific antigens. If the antigens that were isolated from the specific organ of brain or kidney is introduced into another species, then it will act as foreign to that particular species, right? Best example is anti-rabies vaccine. This vaccine contains large amounts of partially denatured sheep brain tissue. When it is introduced into human beings, it induces immune response, causing a great damage to the recipient's nervous tissue. Coming to the other type, that is heterophilespecific antigen. The same or closely related antigens may sometimes occur in different biological species, classes and kingdoms. These are known as heterophile-specific antigens. Best example is Forceman antigen. As I told you just now that these antigens are present in different biological systems. So here this Forceman antigen is a lipid and a carbohydrate complex which is widely distributed in human beings, plants, animals and also in bacteria, but it is absent in rabbits. So it plays a very important role in some diagnostic serological reactions in which antibodies unrelated to etiological agent are employed. This is all about the different types of antigens. Soon I will come up with my next video. Thank you so much.